Hey, this is the video for Gene Expression 3. So go ahead and set up a new entry in your Cornell notes. Remember to start a new page for this video. Check out the essential vocabulary. The vocabulary is protein synthesis, transcription, tRNA, codon, ribosome, and translation. So protein synthesis, remember that synthesis means to put together. So protein synthesis is simply the process in which proteins are made in the ribosome, or put together. So it involves a couple of steps. We have transcription and translation. This is transcription. Transcription is the process in which DNA is copied to mRNA. And this process looks like this. The enzymes are going to unzip the DNA segment. It's going to cut it open. Remember, enzymes are biological scissors or glue. Then they're going to use the DNA to make an mRNA copy. So they will find base pairs or nucleotides and pair up the RNA. Remember that there is no T in mRNA, and they have different sugars, and they're single-stranded. So that once the mRNA has been copied, it will move out and go to the ribosome, where it will make proteins, and then a different enzyme, this one acts as a glue, will zip the DNA back together. Now, the ribosomes are outside of the nucleus. Remember, DNA hangs out in the nucleus. The mRNA goes between the nucleus and the ribosomes. And the ribosome is the organelle where protein synthesis occurs. And we have tRNA, and tRNA kind of hangs out around the ribosomes and in the cytoplasm. And tRNA, you don't really have to know the structure, but if you see this, you might need to recognize it. It's going to be drawn something like this, where you're going to have this kind of weird little shape here. This is different proteins. But then we have these codons or anticodons here, which you don't have to know, and it's attached to an amino acid. The definition, transfer RNA is a nucleic acid that matches codons to attach amino acids in a protein. Now, what's a codon, you ask? A codon are these three little bases, and it is a 3-base segment of mRNA that pairs with tRNA to assemble amino acids into proteins. So this is like U, A, C, and this is like GGA. And what is going to happen is you have the process of translation starting right now. And this occurs in the ribosome. There's going to be two parts of the ribosome, one on top of the other. It basically looks like two halves of a cookie if you look at it from the side. And we're going to have RNA, mRNA, which is down here, and the tRNA up here. And translation is the process in which tRNA molecules use codons to attach amino acids into proteins. This is what it looks like. So you're going to have the tRNA coming in, matching the codon with this tRNA anticodon. And once it figures out and finds a matching pair, it drops off its amino acid. Then the RNA slides over a little bit, and another tRNA comes in and matches with the codons and drops off the amino acid. And this is the start of the formation of a protein. This process occurs thousands and thousands of times to join together a bunch of amino acids. Once these amino acids have been joined together, they're folded into shape, and then that protein will function in a particular way. But we will address that in the next topic. So I would like you to complete this check for understanding, so I can ensure that you know what I am talking about today. I hope that this video was informative. I thank you for your attention, and I will see you soon.